Hi everyone. Excuse my blue tongue. So for this week's Gummy Bear Interactive Lab, I kind of want to discuss what you guys saw in the video. I do have some notes pulled up, so I am going to be referencing them in case you see me going cross-eyed. And this lab was really interesting because while being a chemistry lab and understanding ionization and the different saturations of substances, there's also a biological component to this. And as you can see, I'm in my scrubs. I am trying to get into PA school, hopefully this year. So I really feel like this lab embodied a lot of chemistry as well as biological anatomical processes within the cells and throughout the body. So I do have some notes that I'm going to be looking at just to kind of gather my thoughts. So the gummy bears are made of gelatin, and that kind of represents something like a cell membrane, which is the lipid layer that is the semi-permeable perimeter around a cell. Semi-permeable meaning that only small molecules like water and oxygen can pass into and out of the cell, and the membrane guards the cells from the outside environment, kind of like what we saw with the gummy bears. This retains the cell's organelles, ions, and proteins, and water, which allows our cells to stay alive, in this case, the gummy bear. So what is osmosis? Osmosis is essentially the entering and exiting of water within the cell to kind of maintain an equal concentration both in and out of the cell. Water will stop moving into the cell when there is an equilibrium reached of the same concentration. And we kind of saw how that ties in to why the salted water didn't cause the gummy bear to expand, because the water was so ionized. So what is that molecular compound within the gummy bear? And why did the gummy bear within the unsalted water get huge, which you saw in my video? So my hypothesis was with this experiment was that the water with no salt would equate to a really expanded gummy bear, and I was right. And I think this has to do with the osmotic processes of equalizing concentration. So because the gummy bears are made up of starch, gelatin, and sugars, and gelatin is a type of protein, these proteins are mixed and cooled, and they form the chain with stronger proteins. The molecules are in constant motion, and they tend to move from areas of higher concentration to lower concentrations. Again, this imbalance is what I think caused the water to move into the gummy bear. As I mentioned, the gelatin is kind of like the cell membrane, and the water moves into the gelatin because there are more sugar molecules in the gummy bear than in the water. So salt and sugar have different types of bonding. One is ionic, one is covalent. And this definitely plays a role too, but it's an interesting question to pose as to why one is so much more habited to be drawn in. I think this describes the isotonic, hypotonic, and hypertonic solution. So the salt water would be hypertonic, which means it's hyperconcentrated, and the hypotonic, which would the distilled water was undersaturated. So I predicted correctly, as you guys saw from my video. The green gummy bears that you guys saw were the controls, and it allowed us to compare and contrast the effects of salt water versus no water. So we definitely observed some physical changes, and the effect of osmosis was examined, and over the span of, I think I had them sitting for about 12 hours, more than the experiment suggested, we could see that osmosis was playing more of a role in the distilled normal water. The gummy bear lost a lot of its coloring, I think because it expanded the color dye, kind of those molecules stretched out, making it a lot more translucent. And the higher concentration of salt means that there's a lot less water in the solution as well, and with less water, the gummy bear will end up containing a higher concentration of the water itself. So this is kind of a complicated experiment to explain, so I hope you guys understand. The video, I think, exemplifies it best. So I hope you enjoyed this, and I look forward to doing more videos for everyone. Thank you.